Sciencey person? Okay. All right, so hopefully I'll hit the middle ground here, a little bit for everybody. So we'll see how I do. So first I want to acknowledge Tom Rodhouse, who I've worked with for, feels like generations now, maybe our kids' generations, but, and so I wanted to acknowledge his participation in this work as well. So we'll see if I succeed in convincing everybody to use the beta distribution. So we'll take a poll after to see how well I did. So first of all, what I'm talking about when I'm talking about plant cover is pretty much the aerial projection of the plants onto the ground surface. And so here's a figure from a recent paper that I co-authored with Christian Damgaard, who is a researcher out of Denmark. And this is an important picture to show the distinction between top cover versus aerial cover on the ground. So if we just take a look at this green species here, we can see that it actually has quite a lot of cover, ground cover, but when you view it from the top down, it would actually have 0% cover. So what I just want to point out here is that statistically and ecologically, we need to make sure what we're defining and what we're actually measuring, because it has different statistical properties if we're talking about top cover or if we're talking about the actual on the ground cover. And different field methods align with those two different characteristics. So one of the field methods that folks will use, and some of you maybe have yourselves, is line intercept, where you run a line through a study plot. You might have 10, you might have 5, what have you, and then you're just recording the proportion of the line that's intercepted by a particular species. So this is something I took from Oregon State from one of their field manuals about this particular method. So I actually did this little fun fact when I did my ecology master's degree in clear cuts in Maine. And it was actually that experience that motivated me to become a statistician, because as we're running these transects out, I could see like my partner like over here, and I was like, we're supposed to be straight. So it seems like there could be some improvements in general about how to analyze these data and actually how to do it in the real world. So this is a project that was already mentioned where we've been setting up a long-term monitoring design for submerged aquatic vegetation in wildlife refuges. So here's an example from some field surveys at Lee Metcalf, which is one of the refuges sort of south of Missoula. And so in this instance, they're using a 1 meter square plot, and they're looking down from the top to look at that top cover. So this is the case of measuring top cover of different aquatic plants that are rooted in the ground. As you can imagine, with this particular method, it has all of its challenges because you're actually doing these from a canoe, right? So you're bobbing around trying to measure top cover. So also turbidity is an issue, right? So water clarity can influence your detection probabilities, which I'll get into later. So what kind of data is this? Now I'll start talking to this type of side of the room. So this is an example of a continuous proportion. So a continuous proportion is something that takes on the value between 0 and 1, right? So 0% cover, 5% cover, 10 up to 100% cover is where you get your 1 values. So here's empirical data from that picture that we saw before of sago pondweed, which is one of the, it's considered duck food. So the ducks like to eat the tuberous kind of, they're like potatoes apparently. So it's a really important species to know how it's doing in wetland units, because why do we have wetlands? Well, we're trying to keep the migratory birds healthy and breeding and moving on their way. So this was actually data collected in 2015 and 2016 from Bedoin National Wildlife Refuge, which is up in eastern Montana. And a couple things I want to point out here. So I already pointed out that it's a continuous proportion between 0 and 1. So that's the x-axis. And then you can see there's a lot of zeros. So that means there was a lot of plots out there where they would go in their canoe and they did not record any sago pondweed. Also, there's quite a few plots that had 100% cover of sago pondweed. So you can see that proportion actually changed between 2015 and 2016. And so what happens in between those 2 years is they remove the water from the wetland to dry it down, and that is meant to promote growth of these submerged aquatic plants. So we can kind of see that just staring at this picture that something happened, right? Well, we know something happened. So now we want to see if our statistical modeling can support any sort of inferences about the effects of water management on submerged aquatic vegetation. So what are we going to do with this data, right? Well, based on this little figure, we would not be comfortable, hopefully, using, say, like a normal linear regression model or a t-test or something, one of those things we learned in chapter 3 of the statistical sleuth. So hopefully, if you've read up on some of the literature about continuous proportions, so David Wharton had a paper published a couple years ago where he said the arc sign is asinine. So it was really about continuous proportions. And in this paper, he said you should try a logit transformation. So the logit transformation is the log of the proportion divided by 1 minus the proportion. Basically, it takes that proportion on the 0 to 1, makes it negative infinity to infinity. However, the issue becomes, what are you going to do with those zeros and ones, right? That is not defined in our logit transformation. And so here's an example where I just, fudged a bit, add a little bit, took some away, and you still see like these big spikes, right? They're still there. They're still there. My 0 values, now I just give them some other number, and the ones. So here is the QQ plot. So don't get too hung up on the details, except for you guys. That, what do we want to see? Well, we don't want to see this, right? Like we want to see those dots line up with that sort of faint 1-to-1 line, and you can see that's not happening. So this was the logit transformed data, and part of it, a couple more issues pop out here. So again, the continuous proportion is bounded between 0 and 1. So that basically means you can't go higher than 1 and you can't go lower than 0, right? But that leads to an effect called the floor and the ceiling effect. So you're like bouncing up against those boundaries and you can go no further. And ecologically, this particular type of data stream tends to be skewed, right? So we get a lot of plots with 100% or 0%, and maybe some scattered throughout the middle, right? Another issue with this type of data is non-constant variance. So we'll see a picture of this later. But essentially, the variance is larger around 0.5. And then because of that floor and ceiling effect, like the variance gets really small because you got nowhere else to go, right? So it's just a mathematical property, really. But it manifests in ecological data. So this is why we care about this stuff. So what do? You use beta regression. So there was a recent paper by Duma and Whedon that came out about the same time as our paper. And these are two really good resources, I think, for trying out these methods on your own data sets. And so the approach, again, suggestion is to use the beta distribution. And reassuringly, a lot of plant data sets are consistent empirically with this beta distribution. So here's a picture of Artemisia tridentata, so big sagebrush. And you see this one actually has a pretty nice like unimodal distribution going on here. So this one's maybe not as big a deal as something like this. So this is Pseudoregnaria spicata, where we do see that skewness coming in, right? So we have a lot of plots with very low cover and then some up towards 0.5. So that gives you the skewness in your density. So this is where I convince you that the beta distribution is cool. So for many reasons, in my opinion, but one is that it's like really, really flexible. So for those of you in stats class, it's like, I think the only distribution that does this, in my opinion. So here's a simulation study where we've made up our, perfectly circular plants, because that's how they are in the world. And we have randomly sampled with 1,000 plots, and the plot size is that blue square. And so what happens when you do this simulation is you get this U-shaped distribution, right? So you get lots of plots that fall in the white space, and then others that have fallen completely within the green space. So you get a lot of high percent copper values. So that's funky, right? That nice U-shaped distribution. If we make the plot size larger, so we change the lens that we're viewing the world through, we are going to change that distribution. And now we end up with sort of an L-shaped distribution that has that skewness going on also in the right tail. So the only distribution that really can handle this well is the beta distribution. And so I'm gonna hit something.
Just hit save as, I think.
Save as. And then should I just cancel?
Yeah, or
Save as and then save something.
maybe do reminders.
Where's reminder?
Exit out of that.
Right here. Yeah. Oh, okay.
Okay.
Back in business. Okay, so the beta distribution takes on all these nice shapes, right? Like you can have the U shape and the green, the L shape and the orange, or a nice sort of unimodal but long-tailed. So my collaborator, Christian, there we go, likes to interpret a measure of spatial aggregation, which is actually related to the precision parameter in the beta distribution. It's just a different calculation, where he contends, based on his field work and experience, that it actually provides a measure of the spatial aggregation of the plants. So that previous picture was super clumpy, right? And we made it that way. And so this blue distribution has a spatial aggregation parameter of 0.1, whereas the L-shaped distribution, the orange, has a parameter of 0.3, and then that U has the largest spatial aggregation of 0.5. And so intuitively, if we just think about this a second, so spatial aggregation in plants is like patchiness, right? So it's kind of like a measure of patchiness. And so patchiness implies that you're going to have a lot of white space, potentially, depending on the plot size, right? So it depends on the lens that you're viewing the system. It's important to bear that in mind. But you might have a lot of plots with you're barely missing the patch, or you're completely in the patch. And so that's where you end up with these U-shaped distributions. So back to my example with the sago pondweed. So this was my terrible looking QQ plot. So, you know, I've read some papers, blah, 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 or maybe wrote them. And one option is we could do the fudge factor and say, all right, I'm gonna add a little bit to the zeros, take a little bit away from the ones, and then use beta regression, see how I do. Well, I'm not doing so well, right? Like I still have problems. So the other option is there's actually a distribution called a 0-1 augmented beta distribution, where basically you are assuming that there's different processes happening. So the process, ecological process that leads to zeros is different than the ones dictating the ones, which is different than the abundance measures in between. And so this is the QQ plot of just the non-zero abundance values, and you see that we're doing better, right? Like it's no ideal, but that's pretty darn good with ecological data, in my opinion. And so this, in addition to other lines of evidence, suggests that the 0-1 beta distribution fits pretty well to this data set, to again understand how the water on the wetland is affecting sago pondweed. So there's I think I already mentioned this, but this other paper that came out that I like because I am using this figure, so I didn't have to do it myself, but what they, kind of busy, so bear with me, but what we're looking at is the bias related to estimating the mean parameter, so in this case it would be mean percent cover, using the logit transformation versus the beta. So it's just comparing like, what if I transformed versus use the beta? And then the different colors are the different assumed variances, right? So how variable those data are. And then along the x-axis is the mean. And so one thing I want to point out here is that around 0.5, doesn't matter what you're doing, right? Like you could use the logit transformation, you could use beta, it's probably okay. And so what does that data generating distribution look like? Well, looks like this, right? So this makes a lot of sense because it's basically unimodal, symmetric, you can use the logit, the beta, and you're fine, right? But when we start moving away from that scenario, up here, this is the case where we have a variance of 0.03, and so we're looking at what if I did the logit transformation as that pink line, and notice there's zero bias if I'd used beta regression, and that generating distribution looks like this. So it's highly skewed. So if you see stuff that's highly skewed, you need to be getting a little bit more creative, basically. And so also over here, if we have a low mean, it's like the opposite picture, right? So we have lots of plots that might have low trace amounts of cover, and then a couple that are higher percent cover. So bias is really a function of, if you're thinking about it, like the shape of your data, right? Okay, so how do I do this? Hopefully I've convinced you to consider it. So how you do this is there's the paper that we just had, and importantly, we provide worked examples. So this is all in R, so it's like R programming using available functions. Sometimes we do fit Bayesian models, so there's like JAGS code or STAN code to do some of this. And also this other paper that came out in Methods in Ecology and Evolution has some nice supplements as well. So it's got like additional examples for how to implement these models and the software that most of us use these days. So that's not the end of the story, right? Because there's this other way that people gather plant cover data, which is using Daubenmeier cover classes. So in this case, we still have that 1 meter square plot, and the biologist is looking top down to record percent cover. But instead of assigning a continuous value like 1, 2, 3, 4, 5, they're estimating the cover class, which basically represents a range of percent cover. So there's a couple of common scales used. So there's the Daubenmeier cover class scale. There's also Braun-Blanc-K, the two biggies really. And so in this case, if you, the researcher, for example, if it's like 0 for sagebrush, I'm kind of making this up, then they record a 0. Whereas if it's like between 0 and 5%, they're going to record a 1. So this data type is an example of ordinal data. So ordinal data is basically you have these discrete categories. So it's categorical, but it has an inherent order to it, right? So when we look at this scale, we know that 2 is bigger than 1, 3 is bigger than 2, right? So that's how we define ordinal data. So what do you do with, well, what were people doing with this stuff? They were, much to my dismay, and continue to use the midpoints of the cover classes, right? So they take that cover class, they convert it to the midpoint, and then they use what they know how to use, because that's what we teach them how to use in intro stats classes, is linear regression. So here is an example. This is simulated data again, where basically I have the midpoints of the cover classes on the y-axis, and then a made-up continuous predictor on the x. So the predictor's centered and scaled, right? So it's got this nice going from -1 to 1. So this could be anything. It could be elevation, could be topography, could be precip gradient, whatever. But a couple of important things to note about the scatter plot is, one, we see striping, right? So the striping is because we have categorical data that's ordered, right? And another thing we see is this is a really good example of that floor to ceiling effect that I was talking about before. So if we think about, if you can envision what the residuals would be, right? The variability when it's down at 0 is really super small, like basically 0. Similarly up at 1, but in the middles, like around 0.5, is where you see that band is much wider, which means your variance is really non-constant, right? So maybe I kind of exaggerated this a bit, but I think it shows the point, right? And so when you're fitting like a simple linear regression model to the midpoints, you're assuming that you have this linear relationship shooting through your data. That's the red line. But in fact, it's not. It's curvilinear, right? It's non-linear because it's going between 0 and 1. So you have this bounded proportion. And it's still true even with the midpoints, right? So the midpoints just layers on some more crazy in addition to the continuous proportion. So what should people be doing is they should be recognizing this as ordered categorical data and using the appropriate methods. And so this is really the best way. We're still working on better ways to visualize the data, but what we're looking at here reflects the ordinal nature. So again, on the x-axis, I have, this is real cheatgrass data from John Day fossil beds. And the gradient here is topography. So it's a function of slope and aspect. So we can calculate it and figure out like steep south-facing slopes, flat areas to steep north-facing slopes. And then how you interpret a bar is it's a stacked bar plot. And so for example, at the steep south-facing plots, the proportion of ones is that pink bar. So they're about 50% plots with the 1. And then the purple is the cover class 3. So about 50% had cover class 3. So one thing that's dissatisfying about this is we don't really know the sample sizes going into those bars, right? So you need to be aware of that. Like that could be 2 data points. One was a 2 and one was a 3, right? So that's just FYI. Another thing that pops out is we see that those blues, so that's the 0 cover class, seems to be increasing, right, as we move into steep north-facing slopes. Whereas this is cheatgrass. So whereas in the flatter areas, we have those higher cover classes, so those reds and the greens. And so it is good for visualizing ordinal data and sort of reflects the fact that we have these proportions or cover classes. So the appropriate statistical model is the proportional odds model. So this is an example of a cumulative logit model. Ready, steady, people? This is like the only equation. So that's not totally true, but so the advantage is that it reflects the ordinal nature of the data. It's parsimonious compared to other approaches that you could do. It's readily available, right? So there's like a function in R to fit these suckers. There's textbooks about it. However, a big disadvantage is there is no direct link to that percent cover, right? So when you go to interpret this stuff, it's like really annoying. And so it's hard for me to communicate it to the biologist. It's even more difficult for the biologist to communicate it to the manager when they're trying to manage natural resources. So then we are like, well, this is just kind of dissatisfying. But all along we've been considering this beta distribution because there's a long history for the beta distribution used for plant cover data back through like the 1950s. And it's just cool, as I said before. So it turns out the proportional odds model has a latent variable motivation. So we can envision that how you arise at those ordered categories is by thresholding a continuous distribution. So what we're looking at here is just like a schematic, basically hand-drawn from the Agresti textbook. So this is where this is coming from. And on this far left y-axis, that is the latent variable, so it's y star. And so those alpha parameters are just corresponding to those cut points or threshold parameters. So you can, and then what happens is, so if you're like, say, here, a data point here, you would be assigned a cover class of 1, right? So you can imagine chopping up this continuous distribution, discretizing it into these cover classes. So one thing, so the example that where I've shaded shows you can get at the proportion in cover class 4. So that's the shaded areas in this little dummy picture. And so this might be 2 different years of data collection. It could be like before and after, like whatever, right? So it's just like 2 distinct time periods is what I'm showing in this picture. So that's all well and good, but we already noticed that we're assuming that the latent variable is unimodal, it's symmetric, it's all beautiful, right? And it doesn't provide that natural relationship to percent cover, right? So we're like, well, what if we change that around and use the beta distribution as the latent variable and chop that up instead, right? And so here's an example again, same idea. So this would be similar to, say, if we could observe the continuous percent cover for 2015, and then we chopped it up and put it into the different categories on the right-hand side, right? So this is showing pretty extreme differences in beta distributions. So one's like the left skew, one's the right skew, and then in the middle, I think is the uniform, which is actually similar to the logistic distribution. And the real benefit is it provides that nice interpretation to the beta parameters in terms of mean percent cover. But for those of you that are actually following along here, I already pointed out that we have some problems, and one is the zeros, right? So what are we going to do with those zeros? And in ecology, like zeros mean a lot, right? So zeros tell us about, zeros versus non-zeros tell us about the distribution of species, right? Like where they are versus where they're not can be quite important and could be completely different factors related to where they're going to put their feet down and actually grow and get bigger and expand, right? So back to this cheatgrass example. So here we see like something is happening differently with those zeros compared to everything else, right? And so we have to account for that in a statistical model. So here is where I may lose half of you. So this is the way that I like to explain the basic like statistical underpinnings in of the models that we've proposed. So, you know, we like to be clever, so we call it the OZAB, which stands for the Ordinal Zero Augmented Beta Model. And so in this case, back to like a plot. So if you have, say, any plot I, right? And then this psi parameter is dictating whether or not the species is present or absent at the plot. And then if it's present, then we're going to, you can think of this as a way to also simulate the data. It's kind of similar to how you're going to simulate it or estimate the parameters. So in this case, if it's present, then you're going to draw from a beta distribution and get a true cover value. If it's absent, then that cover value is going to be 0. Now, to get to the ordinal piece, we have to add like another layer. So if there's 0% cover, then we know that cover class is going to be 0. This is the meaningful part. And then this is basically the thresholding part to get. So instead of thresholding the logistic, we're thresholding the beta. to get at these different cover class proportions, essentially. And so this is actually from a Jabez paper that was back in 2016. However, there's another sticky wicket here, which is observation errors, right? So this is an underappreciated aspect, in my opinion, in plant surveys compared to wildlife surveys. So in wildlife surveys, people are aware of the fact if I go out there looking for grizzly bears, I'm probably gonna miss them, right? They're like hiding in the trees, they're in their den, like whatever, right? But they're there, I just didn't see them from the plane, right? And so, and this also happens in plant surveys, particularly depending on your plot size, right? So we're using pretty small plots, but other folks will use much larger plots. And so here what we're looking at is my true percent cover is that red line going across, and then the little dots are representing pretend observers. So say I have independent observers, or I might have repeat visits to the same plot within the correct window of time. And so the visits are going along the x-axis. So we can see these two observations were recorded as a 0 when in fact the plant was there, right? So this is considered a detection error. Whereas with plants, like we might end up having a measurement error. This is probably more likely, maybe, right? In that the true cover is 0.5, but the observer recorded like 0.65, or the observer recorded 0.35, right? So these two different types of observation errors, we need to at least acknowledge that they exist and consider how to estimate them to account for them in a statistical model. So here's back to my little picture. So this is the same ecological process, and now we're just going to extend it, add a layer to account for those observation errors. And so this is exactly what I had before, but now there's another way to get a 0, right? So this would be an example of a true 0. But another way you could get a 0 is if you go to the plot and it's really low cover. So that might lead to a detection error. So it goes undetected, which is going to lead to an observed cover value of 0. So that's an example of the zeros can come about two different pathways, right? Like it's truly a 0 or it's a false 0 because you failed to detect it in the plot. And then again, you might be measuring something with errors. So how we account for that, we basically just add a little wiggle room around the true percent cover, which is that Y notation here on the left, and then account for that in terms of measurement error. So in ordinal data, the measurement error might come across as you're assigning incorrectly an adjacent category, right? So some of the notion about using this ordinal structure is that you lead to less observation errors. But in fact, depending on how variable and your type of plant and the morphology and blah, 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 you might still have issues discriminating like a 2 and a 3 cover class, depending on where that threshold is. So all of these models we have, applied to real data to see how they go. And back to my cheatgrass example. So this one, it is interesting because these things that we see in the stacked bar plot, we actually were able to empirically estimate, right? So we can actually estimate with uncertainty the fact that there was this relationship of more zeros in steep north-facing slopes and higher abundance classes in the flat areas. And then another little nugget of this experience was some of these areas have been burned. And so if you had lumped the zeros in with everything, you wouldn't have been able to detect or really estimate the fact that burned areas were more likely to contain cheatgrass. So most everybody knows this from the news, but in this particular data set, it was hard to tease that out until we had actually appropriately modeled the structure of the data. So I'm really flagging on cheatgrass here, but so again, the same park unit, but they've got multiple measurements over time. So what's interesting here is hopefully at this point, everybody's sort of oriented to these plots. And you notice something happened weird, right? Like something happened between 2011 and 2012, and there was a fire. So there was a large fire across the monument, and we can see something happened. And what's interesting is when you go to fit the beta distribution to this, we do see that the shape has changed, which would suggest that that spatial aggregation has changed for cheatgrass after the fire came in. So at first it starts out as this U shape, pre-fire, and then post-fire, it turns into that L-shaped distribution. So again, in 2015, at the same park unit and some other units here locally at Bighorn Canyon, they did some really nice double observer work. So they had 2 observers go out there to each plot and get repeated measurements of the cover classes. And this is results for 4 different species in bare ground. And bare ground's fun to play with because it tends to have the most observation errors, frankly. And so this is just showing ignoring observation errors. And then if we account for those observation errors in the blue, we see that really bare ground's the only one that seems to have shown a shift. So this was reassuring to Tom, who is the one that participated in this, because it basically says it's probably okay to ignore the observation errors in this case. But we wouldn't know that unless we had fit the model, right? But what is consistent with all of the simulation work we did is that if you were to ignore the errors, what you'll see is for the bottom panel, so that's the probability of presence or the probability of the species being there, you're going to tend to underestimate the probability of presence and overestimate mean percent cover. So intuitively, if you think about that, so if those plots that you go to have really small trace amounts of the plant, you're likely to miss it. So you're gonna underestimate how sort of ubiquitous it is. And then it's almost like you're only recording those plots that have higher percent cover. So that's why your mean is overestimated. But luckily in the empirical data, we didn't find much evidence of that. But in some cases, it could be a big deal, right? Depends on plot size, shape, configuration, what you're measuring. So how do you do this? So this is another recent paper. And again, we have the code available for folks to play with these models. And all of these examples in the paper should be, hopefully, reproducible using all that code that we've produced. And with that, I will take any questions. Cool. Christian?
Okay. First of all, that was awesome. Very interesting model and a cool idea. My question was about the Ozabe graphic that you had up there.
Mm-hmm.
When you're estimating that sigma squared and that normal distribution for the errors, can you use that estimate then to talk about like the magnitude of these observation errors? Or
Yes.
Yeah.
So that's a really good, very insightful point. So in the paper, we actually show how by tweaking that variance parameter, you can basically like increase it's related to, so in the empirical data, most of the discrepancy was like among one cover class, right? So somebody recorded a 2 and somebody else recorded a 3. And so it is correlated, like related to that sigma, the measurement error piece. And so in that paper, we have a figure that sort of shows 2 examples, like if it's a 1 versus a 0.5, and you're just basically shifting the probability of being in the wrong class. Yeah. So it should be related to, again, you have to have those repeated measurements of the discrepancy in the cover classes between the observers to estimate that thing. Good question. Christian again.
And I thinking back to the proportional odds models, you have to assume the same relationship between the predictors and the response across all of the categories. Is that also true of the Ozabe and Ozabes?
No. So right, so that is one of the inherent issues, right, with the proportional odds assumption. So that is the proportional odds assumption. And so in, I think the Jabez paper, maybe in the me paper too, like we provide sort of ways to assess whether or not that's a problem. So in the Cheatgrass example, where we saw something happening differently with the zeros versus the other one, you can actually see that in the diagnostic plots because we know that that relationship with topography is different. So you actually kind of, so in that case, we were better off doing a hurdle approach even with the proportional odds model, where like from the cover classes 1 through 6 or whatever, it was probably okay to assume a similar relationship, but the zeros were definitely doing like something else. Does that answer your question? Yeah. All right, Rob. let's see if somebody whose name I don't know will ask a question.
I'm just thinking about all the things that confound the data that you're operating on here.
Mm-hmm.
And so we have to believe going in with the analysis that the original sample design adequately reflects the population, the spatial representation, and all these other considerations,
Mm-hmm. Mm-hmm.
even before we start to see what the data is telling us. So
Right.
can this method be used at all to corroborate some of that initial design work and whether we are being representative in what we're sampling?
Not really. So statistically, all of the assumptions around the survey design are what allow us to make, in your cases, right, inferences to that larger spatial unit, right? So in the National Park Service, they'll have these 1 meter square plots, and then they have a defined sort of larger polygon. Maybe it's a unit. Like in the wetlands, it's easier because it's like, wet versus not wet. But in these other places, it might be based on like other information like fence lines or roads or whatever. And so when we do a random sample within that, that allows us to make inferences to that unit. But would this approach allow us to, say, confirm that the plot size is correct? Is that that's another aspect, right? And I think the double observer work did, in a sense, that we were like, oh, this is great, right? There's very little evidence of observer errors. And my suspicion is if we had increased that plot size, so a lot of the work that's out there that looked at observation errors in plant surveys, they have like 10 by 10 meter square, like much bigger area, which increases your chances of missing something, right? So, so I think, kind of from the side, it provided reassurance to everybody, right? That we don't have real strong observation errors when we're doing this stuff. And whether or not, I guess I was gonna say, as we dig deeper into the patterns in the data, like assessing whether or not we are capturing any gradients of interest is really where those questions will be answered, right? So in Christian's work, for example, as a small subset of he's done work in Grand Teton with their data. And one of the ideas is like, well, are we capturing gradients so we can talk about climate change? Well, the answer was kinda. I mean, so meaning there was like not a lot of variation to date among years, and spatially not that much either, but maybe that is representative, right? Like it's hard to say, but
that's why long-term monitoring is crucial.
That's right. Mark?
Yeah, okay. Yeah, so that was really interesting and definitely a problem that I see a lot in students' projects. And, a lot of people around here have data like that. So for, say, my 512 students, are you saying they should be reading those appendices and trying that for their project? And in second part, if they get stuck, can they contact you for help?
So the answer is yes, if they have continuous proportions or ordinal data arising from a continuous proportion that they're interested in. And of course, I live right down the street. That offer goes to anybody in the audience.
Thank you.
Contact you before we start.
Well, yeah, in this case, sometimes it's, yeah, ideally, right? But,
I'm not sure whether I'm on here or not, but what's an alternative to using cover data? So we can look into a plot and we can get a perfect species list, but you mentioned a couple of kinds of cover we might measure. Different plants are so different. The wind is blowing or not, I'm tired or not. Greg Smith, back whatever that was, 1965, reviewed so much of that I feel is not discussed. And I've been out estimating with people that I highly respected, hopefully the same, and we'd be 3 and 4 times off.
Yeah.
My question, I'll just restate it. What's an alternative to using cover?
Well, so my collaborator, Christian Damgard, would claim he's a big fan of the pinpoint method, right? Where you drop the pin.
Exactly.
Yeah.
What a pain in the neck.
Exactly. So I didn't mention that because it seems super impractical. So what I would say is you record your various measurements, and then we can actually account for the fact that everybody's kind of off from each other and still get unbiased estimates of the parameters we're interested in. So if anything, these models are basically saying, we get it. Humans are different. We all have human observation error, and it's okay, because if you have independent measurements, now we can actually account for those in the statistical model and still get at the right answer instead of ignoring it and pretending it's not a problem, right?
So I have some plots that I've sampled repeatedly for 25 years and some other that I've sampled repeatedly for 50 years. And I wonder if I'm the same all the time.
I can answer that. No way you can. No.
I keep getting better, right? But still, it's different,
Yeah.
whether I'm better or worse.
Yeah. Christian again?
Yeah,
It's okay.
just a question about your opinion on this sort of thing. Do you think then that when possible, you should be hitting these plots twice to be able to estimate that measure error, or based off of what you saw in that empirical data, it's...
I think it, now I'm going to pull the statistical thing, right? It depends. So like these projects, well, Aaron and Rob could better describe this, but there's a lot of training in the beginning, right? So they try to calibrate observers, and particularly with the Fish and Wildlife Service, you calibrate both your identification and your observers on cutouts. So they'll have like these 1 meter square things, and then they'll have like cardboard cutouts to calibrate before they even go out in the field. So there's that. There's also the size of the plot and like the types of plants that they're looking at matters. So I could see how for other systems, it could be a bigger problem, right? And so some of the like grasses, for example, I mean, so the pictures I showed was like sagebrush, like kind of around here, I'll know what that looks like. That's kind of a hard one to miss. Like I can identify it, right? So I think it depends on what it is that you're looking for, and also like the importance of your outcome. So if you're trying to inform a manager, like help them answer a question, and if you tell them the wrong thing, there's like a huge consequence. I would feel better if everybody's like accounting for these potential errors that could lead to the wrong conclusion, right? So I think it depends on kind of the decision that you're making and the system that you're working in. So the context matters.
Can I add something? So Kathi came out in the field with us. I work on white bark pine, and we've been getting slammed by some other people in the white bark pine world about how we were canker counting on these trees. So there's these infection sites on trees, and she and Wilson Wright came out and they watched us survey the trees, and they had this aha moment that I never thought about. But because we canker count, we spend a lot of time on each tree, and we're really just, presence absence is really important to us. It's not necessarily are we matching up the same number, but I think the way you approach whatever you're studying, if you have some sort of backups like that, it helps. So think about, you might want to spend more time or I don't know. I mean,
Yeah.
There's things like that.
Yeah. And so another researcher was like, for the plants, you could do time to detection. Like, okay, I'm looking, how long did it take me to find it, right? So you could blend sort of those time to detection models for wildlife surveys into the plant world, is another idea. If anybody wants to do that for, no, I'm just kidding. For a chapter of something.
Just to follow up on your question from before, Christian, I think independent observations, periodically at least, also go a long way just to document quality assurance of your overall data collection effort and all the investment that goes in that. So data quality, data assurance is a consideration for all this work, especially work that has such weirdness as ecological parameters.
Right. And so for like, I kind of alluded to this, but for invasive species, like when you want to get it fast, like when it's sparse, like that is probably the most critical piece, to have these multiple observers to actually get at detection errors, just like you would for an endangered species or whatever. So
So the timing. Oops.
That's okay.
I just
Go,
raised
and
my
then I
hand.
see Mark after Aaron.
Yeah,
Go ahead,
because
go ahead.
for some of the plant stuff, if we went too early, the phenology is really important. So you really need to be consistent with when you, and like last year was really wet or cold, so you can see some shifting just based on that. So you have to keep that in mind too. What's it doing seasonally?
Mhm.
So it kind of confounds it. Yeah, or
Yeah.
too late. Yeah.
Yeah, that's a really good point. Mark?
Well, that kind of brought up another question, or why not just turn it into binary presence absence and avoid all these issues of what the percentage was that's hard to model, kind of?
You could. I mean, that's always an option, but there's a lot more information in these abundance, right? So if you're out there like taking the trouble to actually record some measure of abundance, proxy of abundance, you might as well use it.
Yeah. Or you could measure a lot more sites if you just look for presence absence.
Yeah, there's trade-offs again. So
Yeah.
it depends on like what the questions are, and particularly for like trend detection, I think abundance is a more sensitive measure of trends over time versus presence absence, right?
Yeah, monitoring like the impact of climate change, right? Sagebrush is going to hang around for a very long time.
Right.
Do you want
Yeah. Yes.
Correct me if I'm wrong, but the first part of the zero portion of that is actually doing that.
Yes.
Right?
Yes.
Like I've worked with zero fitted Poisson models.
Yeah.
I think it's, I'm definitely not a statistician, but I think it's pretty similar.
Yeah.
It might, thinking of actually about the white bark pine thing where you're actually counting tankers might be more appropriate. But yeah, I think you're getting both.
Yeah, exactly. You understood my diagram. So I'm happy, like my email is not there, but it's around. You can Google me, so I love talking about this stuff, so it's not a bother.
Any final questions?